The fourth and last presentation for this slot. Then we have a short break so you can ask more questions and get into discussion. The fourth company is Lumatics Biotech. It's a startup from Garching, and their concept is a light-controlled affinity matrix as an alternative to protein A chromatography for antibody purification. As I work with it, I'm very looking forward to see what the alternative will be. And Andreas Reichert and Ingmar Polle will present.
Yeah, thank you for the kind introduction and good morning, everybody. I'm Andreas, and this is Ingmar, and we're going to present now the technology of Lumatics Biotech. Yeah. Since we've been experimenting with light since the discovery of fire in the early Stone Age, so that's no wonder that one of the fascinating innovations are about light. Just imagine, light changes the way we live. What would life without having a light bulb? It was developed over 250 years ago by Thomas Edison. Light changed the way we communicate. Light changed the way we work. And now, since last year, light changes the way we purify antibodies. As you may notice that the global pipeline for biologics has diverged in the last years. If we could have a look at the sector of antibody, which is the biggest part of it, here we see a rise over 12% over last year, and there are very diverse molecules in pipeline. For example, bispecific antibody drug conjugates, MAPs, FAPs, and other entities. As a reaction of the coronavirus pandemic last year and the year before, the biotechnology industry just had to finish 5 antibody molecules, which are approved and already marketed, 55 in phase 1, 2, 3, and 36 are already researched. This device here, you're all familiar with. I think everybody of you in the morning use this device to make this event as safe as possible. So we use this device to fight against the corona disease. It's just possible because we have an analytical antibody covalently immobilized on that device here. But purification of antibody, it's very hard and difficult. So the company or customer has to purify the antibody from a crude extract, and this is always done by affinity chromatography. And affinity chromatography is basically 3 steps. The first step is bind, wash, elute. These 3 steps especially are 3 steps which make the company Kiergan big. So the target molecule is bound to the matrix, impurities are washed away, and the target molecule is recovered by applying elution conditions. So having an affinity chromatography in a downstream process gives the opportunity to reduce the number of purification steps and therefore increase production yield by reducing and reduces actually the bioprocess development time and in essence reduce the time to market and overall reduce the costs. So, but protein A chromatography was established almost 50 years ago. And this technique is made for this molecule. This is monoclonal antibody. But today it's completely different. We have fragments, we have antibody drug conjugates, we have radiochelators, we have bispecifics, and so on and so on. And all these molecules are very sensitive. And they are all especially sensitive to low pH. And this is the problem with the protein A chromatography. We have the low pH to elute the molecule from the column or from the resin. We have other problems like high content of aggregates or very complex purification procedures or an absence of a protein A binding site, so we can't use standard protein A resins. So this is the point why we definitely need a new technology to purify antibody and especially antibody modalities. Therefore, we at Lumatics Biotech developed a completely new platform to develop a light switchable affinity matrix. We start with a ligand, could be any ligand. Then we apply a molecular photoswitch to the ligand. We have a compound pool that's over 16 photoswitches in it. And this is what we call the optically controlled affinity ligand. Via a linker, we can attach it to a solid support. Could be a membrane, could be a bead, could be a magnetic particle, whatever. And this is the optically controlled affinity matrix. In a standard Lumatics purification process, we are, in the case of the antibody purification, we apply the supernatant to the column, and the column is illuminated with red light. This means the column is in binding mode. Antibodies can bind to the column. We will wash impurities away, stay in the same buffer, and then we apply blue light. And the column is switching from binding to desorption mode in a few seconds. Then we have the antibody in a pure manner in the buffer of choice, mild elution. You can run water, you can run PBS, whatever suits you best. Regeneration is fast as a blink of an eye. In just a few seconds, we apply red light, and the column is ready to go for the next purification cycle. With our Lumatics USP, we have an increased product quality due to the mild elution. We have increased productivity at reduced production costs because we save time, we save buffer consumptions, and we are economically friendly. And we are ready for multi-column chromatography because of the very fast switching between binding, desorption, binding, desorption. This is our vision. Our vision is to have a new tool to replace or even compete with the protein A capture step used by pharma downstream. When we look at the market, we can see that the market is valued over 600 million in 2019, and it started to grow over 1 billion in roughly 10 years. We have a market share in Europe over 21%, and we already identified over 4,000 customers in academia and over 7,000 customers in the industry. The market for antibody drug conjugates are even better. Here we have 5.8 billion last year. And just keep in mind, here we are not a competing technology. Here we are an enabling technology. We have a lot of customers, they ask us, can we have your technology because we cannot make it with our standard protein A procedures. This is the pipeline at the moment. We are already near market entry with protein A, G, and L. We are at TRL5 with our Pure Light strap, so that's a strapped avidin variant for binding a biotin, light switchable. And at the moment we are planning to do a light switchable nanobody as a new platform. So we are try to make a nanobody scaffold light switchable. Innovation is not just designing and new products or producing products. It all starts with a great team, with people who are very like to transform the ideas into business, into real business, and to real products. And this is our team. This is just the co-founders. At the moment, we are a team of 4 embedded in a very, very strong network. So we are happy now to answer all your questions, and hopefully we can improve or boost your purification process. Thank you.